So today we are going to talk about morphology of cells and how we can make observations about them when looking at stained or unstained images of our cell cultures under a microscope. So to do this, we are going to start off by looking at some morphological markers to define different types of cells. A typical normal healthy cell, normal is kind of a misnomer here. It doesn't have to be normal as a completely healthy cell. It could be a disease, but a cell that follows typical rules of cell regulation regarding cell division and replication. That's the type of normal we are talking about. So a cell that is undergoing cell cycle as usual is going to show up as one of three different ways. In the normal setting where it is not undergoing cell division, you would see it as a membrane, bound nucleus. The nucleus is going to be small inside it. The nucleus will take up no more than 40% of the total cell area or volume. So you will see plenty of cytoplasmic space. Now I've drawn a circular cell membrane, but that's not actually true, right? Cells are going to look different depending on what type of cells they are. But you would see some type of a membrane around it, and then you would see a nice clean nucleus inside. The nucleus shouldn't be misshapen. It should be more or less round, a circular nucleus. Inside the nucleus, you will actually see dark, tiny dots, and those are your nucleoli, and I'll show those to you later on. The nucleoli in a normal cell are typically just a single or two nucleoli per cell or per nucleus. Then you can also see cells in a typical setting that are undergoing cell division. A cell that is undergoing cell division and is in the metaphase stage will show the chromosomes lined up along the metaphase plate in the center of the cell. You won't see any nucleus in this case, obviously, since the cells are in the middle of cell division. And finally, you could see the cells in late cell division when they are undergoing cytokinesis. You would see two equal daughter cells forming with two equal sized nuclei inside. So that would be a normal cell division happening at that time. Now, sometime either because the cells are damaged or diseased, it could be because they have mutations that are leading to dysregulation of cell cycle or the way the cells are dividing. It could be because they were exposed to toxins or UV light or something else that disrupted their normal function and have damaged them to the extent that they are unable to do proper cell cycle. In that case, the cells are going to look different under the microscope. So there are three different ways that we can find these cells. In the simplest of these states, you would still see a cell membrane similar to how you saw in the normal cell, a typical cell. However, this time, the nucleus inside is going to be pretty large. It's going to be significantly larger than a typical cell, and it is going to sometimes even take up almost the entire space of the cell inside the cell membrane. So it is, by definition, we usually say if a typical cell shouldn't have a nucleus that takes up more than 40% of the total cell volume. Stalled or arrested or somehow disrupted cell is going to have the nucleus take up 60% or more of the total cell volume. And so if we look at the cell, again, I'm showing you an exaggerated version. This nucleus is so large that there's almost no space, cytoplasmic space left behind. The reason for this is that many times when cells get disrupted or when cells get damaged, they're still able to replicate their DNA. And they're still able to go through the synthesis phase. So now, instead of having the typical diploid cell, so a 2N DNA content, you have cells that have 4N DNA content. And that results in a larger nucleus. Sometimes, as they become stalled and unable to proceed further, they go ahead and go through the synthesis phase again. So now you have, instead of a 4N, an 8N cell, DNA content. So this is a multiploid cell, and that can continue sometimes even up to 16N stage. So that would cause these cells to have a very, very large nucleus with very little cytoplasmic space. This is a typical, what we call a stalled or an arrested cell. You can also have cells that were maybe not so damaged that they got stalled in this kind of, middle of cell synthesis or end of synthesis phase, but rather went a little further and attempted cell division. Sometimes they were damaged in a way that they are no longer able to form a nice clean metaphase plate because the DNA content, DNA is damaged or is not equal, the chromatin is mismatched, so the chromosomes can't quite line up the same way. It could also be that they were treated with a drug like Taxol that we talked about, which is an antimitotic, and it works by disrupting the microtubule depolymerization, so it kind of freezes them. So microtubules, since they are the ones that are making your spindle fibers and are responsible for the spindle formation and the poles and the lining up of chromosomes on the metaphase plate, if that's disrupted, the chromosomes will have no guidance system, and you will end up with chromatin kind of getting condensed and then lining up randomly or forming all these kind of multi-poles, attempting to divide but not able to do that. This would be another example of a stalled or an arrested cell. Lastly, you can have cells that are even further in cell division. They've attempted to somehow segregate the chromosomes, divide them up, and attempt the cell division. But now they are unable, because they didn't do it equally, they're unable to make equal daughter cells. So you'll see one side or one of the daughter cells that is forming significantly larger than the other. Again, my depiction is exaggerated. Sometimes you see them just like that. Sometimes you see it where the membranes look pretty similar, but the nuclei are very different in size, indicating that the chromosomes did not segregate appropriately. So that would be another example of a stalled or an arrested cell. Finally, you can have cells in your culture that are dead and dying. Now, that's going to be present regardless of whether you have a normal cell culture, a typical cell culture with no issues, untreated cells, or if you have cells that are actually diseased or somehow damaged or treated with a drug that may cause that to happen. You will always have some dead and dying cells in there. Now, these can show up in multiple ways. If you're looking at cells that are still in culture, still in media, they may show up as floaters. If you're looking at stained slides of cells, so you've removed the floaters, they will show up as really dark balls, stained balls, 3D almost looking balls. And these, the difference between this and a stalled and arrested cell is that there is no nucleus inside. So this is just a dark blue or a darkly stained cell membrane, essentially, with nothing inside that you can see clearly. So the nucleus is already disintegrated. That's one way that it can show up. These balls are typically much smaller than a typical cell because they have gone through enough of destruction in the process of dying that what you've left is a small cell body. You can also see them at an earlier stage undergoing apoptosis or programmed cell division, in which case you'll see kind of bubbling on the cell membrane and little blebs forming, and the chromatin inside may be getting fragmented, the nucleus inside may be getting fragmented. This would be an example of an apoptotic cell, a cell dying through programmed cell death or apoptosis. You can see this process later on in a later stage as just little remaining blebs and cell bodies, remnants that are present in your stained slide area that you're observing. Finally, you can see cells that are undergoing death through other mechanisms. Some of those we'll talk about are necrosis or autophagy. In both of those, one of the things you may notice at some point are presence of vacuoles. They literally look like there are holes in the cells, and I'll show you some examples of that. But that is an example of cell that is undergoing death soon right, it's imminent death or it's going to happen pretty soon, and it is just going through the early process of that when you see these vacuoles forming. At a later stage, if it is a necrotic cell, the cell would be expanding in size. So it will be a really large cell as it gets ready to burst and get destroyed. And if it is autophagy, the cell would be shrinking in size and becoming smaller until there is almost nothing left in there except the nucleus. And at that point, that cell, it does not have enough content left over to continue its survival, and it will eventually die out. Now, autophagy can also be a survival mechanism. It lets them last as long as possible, waiting for the conditions to improve, waiting for the environment to get amenable for survival. And if at any point during that initial stage of them ingesting themselves and going through autophagy, if environment improves, if conditions are good, they can revive and they can become a typical normal cell again. However, there comes a point of no return, obviously, where there's just nothing left over. The cell contents are so damaged at that point that they will eventually die out. Now, because we are many times when we are working in our class, we work with cancer cell lines because they are easier for students to handle. There are some additional markers that we look at for cancer cells or transformed cells. The transformed cells are going to be a little bit different than our normal cells simply because the whole idea behind a cancer cell is that they have dysregulated cell growth and dysregulated cell cycle due to mutations present in the DNA. So a typical cell or a normal cell will have lots of cytoplasmic space like we talked about. All the cells in a normal culture will look very similar. There'll be some variation in size, but overall there'll be, a small deviation. And they will all have a nice clean nucleus, similar in size to each other, and they will all have a single or maximum 2 nucleoli. So here you can see those nucleoli. And those nucleoli will be very small, very concrete, uniform circle, and they won't be misshapen. And you will see a lot of fine chromatin in there, obviously, because they are going to be going through typical cell division signals. In the case of a transformed or a cancer cell culture, what you see instead are cells that are very variable, depending on how mutated they are and what type of cancer it is and how aggressive it is. You will see cells that have a large variation in size and shape. You will see nuclei that are misshapen, that will not look like typical round, nice, clean nuclei. And you will have lots of nucleoli inside, where they will show up as multiple circles within a single nucleus, and those circles may be large and misshapen in many times. If they are undergoing cell division, just because they have so many mutations, many times they end up with arrested mitosis, either because there are just too many chromosomes to segregate and divide, or they are unable to create the metaphase because there are so many mutations and they create this multipolar mitosis where they are unable to divide equally. You can also see sometimes lagging chromosomes within a normal typical cell division as it is happening. Another thing you may notice is that these will end up with multiple large nuclei. So not just nucleoli, but actual nuclei. Sometimes a way for them to survive and get out of this multipolar mitosis is by making multiple nuclei, actual nuclei, within a single cell membrane. And that would also be an example of a cell that can no longer obviously go through cell division because there's so many nuclei inside. So it's in a way a stalled, arrested cell, but it's a multi-nucleated cells. And that is a common observation in high-grade cancers and in certain types of treatments. So again, finally, just reminding you of the different ways cell death can happen. You would have a typical cell, a normal cell. If you treat it with some kind of a damage inducer, whether it is UV light or gamma radiation or some kind of a toxin in form of a drug treatment, the DNA gets damaged, the cell gets damaged, and depending on how the cell is damaged, whether it is damage overall to the cell membrane permeability or the nucleus or the mitochondria, whatever it is, it will respond differently. It can, as a response, go through apoptosis, which is programmed cell death, and this again shows you the blebbing and the little blebs coming off as the cell destroys itself in kind of a suicide, essentially. And in this is a very clean death, so it only kills the cell in question and nothing else around. It could go through autophagy, which is where it consumes itself until it can find enough resources outside to safe resources to continue its survival. And if it doesn't happen and it consumes too much of itself, it eventually dies. So you will see these vacuoles forming, and then eventually the cell will shrink to a point of no return where there's just the nucleus and the cell membrane. Or it could go through necrosis, where the membrane permeability is damaged, there's an influx of fluid, a large vacuoles form, the nucleus gets destroyed, eventually the cell bursts. This is a very explosive death. It damages other cells, even normal typical cells around it as a result. So these are some of the things that you can look for when we are observing slides. Now let's look at some example slides and see if we can find some indication of these, some markers of these in actual stain slides. The slide images I'll show you are mostly images from student slides from the previous semester. So these are slides from students just like you doing their experiments in class. Okay, so in this first picture, so this particular image is from a HEPA-RG cell line. These are transformed cells, but they are normal untreated culture. So in this case, you see the cells are completely normal, typical looking. They have lots of cytoplasmic space. They have nice clean nuclei inside. You will see multiple nucleoli sometimes inside the nucleus because they are transformed. However, these are typical cells, untreated cells, so they are behaving the way they normally should. They are going through cell division just like they would in any other case, and they are dividing as usual. I've drawn around some of these already for you, but I'm going to show you a couple more. So here is your cell membrane. Inside you will see the nucleus, and then you have the dark concentrated nucleoli inside. And like I said, you may see multiple nucleoli since they are transformed cells. However, these are typical, what you would consider normal cells. They are not stalled, they are not dying, they are behaving the way they should. Here's another one that I'm drawing on to show you another example. And there was the membrane, the nucleus, and then the nucleoli. So if we were to look at this particular one and measure the nuclear diameter, we would be looking at, or body diameter, we would be looking at this entire length, all this fuzzy stuff. That's the cytoplasm, the membrane. If we were looking at nuclear diameter, I would be looking inside on this nuclear membrane, and that would be my nuclear diameter. So that's how you would differentiate between those two. And then again, this one only had 2 nucleoli, and those were pretty nice and clean, so that was a good normal cell. So if you look over this culture, majority of this is containing typical normal cells, and I don't actually see any dead or dying cells in here, so this is a very clean, untreated culture. Next, we are going to look at another example. At the zoom, I don't need a zoom, 1 second. Okay, so here's another culture. This particular one has been treated with Taxol or at some low concentration, so we could still see cells that are there. They're not all dead yet or dying yet. And now you can see cells are looking different. And in this case, you see again that you have your cell membrane outside. And in many of these cases, you're going to have stalled cells. And you see that in here by having the nuclei right inside. Now, many of these actually have multinucleated cells. So in this case, you see 3 nuclei in the same cell membrane, maybe even 4. And you will see when they're multinucleated, they are going to have nuclei of various sizes. They're not even going to be the same size nuclei because it depends on how much DNA content is inside those nuclei, individual nucleus, right? So there's a single cell membrane, and then there are 4 nuclei within this, each one of them containing its own nucleoli, right? So here is one nucleoli, this one has its own, this one has its own, and there was another one. Here's another one. This cell is actually undergoing cell death eventually. You can see the vacuoles forming in there that look like holes. You can see these are the vacuoles, right? They look like holes in the cell. You see the nuclei are getting disintegrated. They don't even look like real nuclei anymore. And you see the cell membrane, right? Oh, that looks like a monster. That looks like Shrek. Oh well. So that would be an example of, again, an arrested, or this one would be a dying cell. But majority of these cells, if you look at them, they are going to be either dying or stalled and arrested cells. Majority of them were stalled and arrested, and then obviously some of them are dying. This one has no discernible nucleus inside. It's going to be dying, or it is dying at this time. So again, that would be an example of what you would be looking at. Here are some more examples. Again, this is a multinucleated cell. There are 2 nuclei. There's a single membrane. This would be what you would call a multinucleated cell. And then you have some cells which are clearly in trouble, but they're still looking like a typical cell as such. When they're nice and round perfectly like this, they are in trouble of some sort. They're mostly going through autophagy or getting ready to stall and arrest or die. Okay, so let's look at another example. In the last image, you had mostly stalled or autophagy, or cells undergoing autophagy. In this example, on the other hand, you have mostly stalled cells, or you have cells that are either going through necrosis or apoptosis. You can tell that because here are some, stalled but otherwise normal-ish looking cell, right? Otherwise typical looking cell. So here is a cell membrane. This actually has multinucleated cells, so there are multiple nuclei, tiny nuclei within another membrane. But this one has a typical size and some nucleus inside, and you can see that a lot of these are stalled, right? This one is a very good example of a stalled cell. So here is the membrane, and then there's a single large nucleus inside with a single nucleoli inside. So that's a stalled cell. Now compare that to some of the other cells in this culture that have these big vacuoles, the holes inside them. Some of them are super large, right? So here is a cell that's super large in the membrane. And then you have all these vacuoles inside. This is a cell that's going to, and you see some pixelation in the membrane, and that indicates the membrane permeability is compromised. Here again, you see some pixelation in the membrane, indicating that there's membrane permeability is getting, going away. And that indicates that these cells are going through necrosis as opposed to autophagy, where the cells were going to become smaller and smaller and very nice, round, compact state. Here is one that may be already exploding almost. If you look at it, you see, like there's all this faint exposure on this side with some vacuoles, and then there's a nucleus on the other side. Other cells in here, like hair, which has a lot of blebbing and is round, is and more undergoing apoptosis. This one could be undergoing apoptosis or necrosis. It's a little bit in between, but clearly a lot of these have the typical features of necrotic cells. So these large holes or vacuoles in the membrane, along with pixelation in the cell membrane itself. So in the cytoplasmic space, you're seeing these big large vacuoles, empty spaces, and then they're not empty, they're fluid filled, but they look like they're holes. And then pixelations in the membrane indicating permeability is getting lost. Membrane permeability is compromised, indicates that these are necrotic cells. So that would be example of those. I just thought it would be super interesting to see, just because look at this cell. This is a necrotic multinucleated cell. This cell is going to die through necrosis. It's going to be explosive, and some of it may have already happened, as you see lack of cells around it. So it may have already killed some with the contents getting spilled out. That is a humongous cell compared to all the other cells that you see in that culture. So this would be one of those exaggerated examples where you see lots of nuclei completely getting hammered and destroyed inside, lots of vacuoles forming, and the membrane getting larger and larger as it gets ready to explode. In the same culture, this seems like a very aggressive culture. You see some cells that would qualify for typical cells still, right? Here is an example. This is a single membrane, single nucleus, nucleoli inside, and then here's another one. With the nucleus, nucleoli, typical cell. Other cells in here are clearly stalled. Like here, this is a large nucleus, right? This is a super large nucleus. Sometimes some cultures are kind of, sticky. The cells are very close to each other. It's hard to tell them apart, so you have to pay a little harder attention on where the membrane of one is compared to the other. But this one is clearly a stalled cell, very little cytoplasmic space. And here you have some multinucleated cells. So you can look over here and I can see my membrane, and then multiple nuclei inside. And those nuclei are terrible. There are so many and so variable in size. So you can see that this would be definitely a transformed aggressive cancer cell line that would result in this, or some really bad treatment. So there is an example of a multinucleated cell. So multinucleated, a typical normal cell, a stalled cell, and a necrotic cell, all in the same slide big frame. Here's another similar slide that contains a different cell line and treated with some particular drug, and you can see all different types of things happening in this one as well. So you see some cells, these are large cells in general, I can see that. They have nice membranes, they have nuclei inside. Now some of those nuclei are large and some are small. So here would be a typical cell. You can look at this one and you see the nucleus and you see the nucleoli, and so this would be a typical cell. And then you have another typicalish cell, on this side, but then you have stalled cells, and this can be an interesting cell. It almost seemed like it had a mini nucleus inside as well. Here's another example of a stalled cell. So there are some very large nuclei inside. And then you have some cells that are going to go through necrosis. Here's a multinucleated cell with lots of empty vacuoles. And right now the nuclei are still intact, so I would just call it a multinucleated cell. But because of the vacuoles, you know that the cell will eventually die through necrosis. Here's another typical cell, lots of cytoplasmic space, nice clean nucleus, and then your nucleoli inside. And then you can look over here and you see some dead bodies, remnants of cells that have undergone apoptosis. Here is another one that is going through apoptosis right now. You can see the blebs forming on all over the place. So you have some examples of apoptosis happening cells, cells undergoing apoptosis and dying through it. You also see some dead bodies kind of scattered here and there. So some cells clearly have already died through apoptosis in this particular slide. So that would be another slide, a typical example of culture slides that you may be looking at if you were doing these treatments in your class yourself. Last example, sometimes we are looking at cells that are maybe of neuronal lineage. So we work with mice and 2A neuroblasts sometimes, and we try to differentiate them into neurons. In that case, we are not only going to be looking at the cell bodies, but also these processes that are forming. And we may be measuring their length or how many times they are dividing, or once they are, so if they are just clean single or if they are actually dividing further to make connections. So there'll be other morphological things that we are going to be looking at as well. So microscopy is not just used to look at, hey, there are some cells, look how pretty they are. You can actually make lots of different types of observations from them. And you can look at lots of different things, just morphologically as well as measurement quantitatively from them. You can look at number of nuclei, number of nucleoli, presence, absence of vacuoles. You can look at how the cells are dying, how well the culture is surviving a particular treatment, if they're differentiating or not. So lots of things that can happen. It's simply by looking at these slides. For your assignment this week, you had worked on some slides using image analysis, and you had made some observations similar to how we make in the classroom using our microscope. And for your slides, what we had looked at was total cell number, and then you had categorized the cells into normal, stalled, multinucleated, and dead and dying cells. And then you had looked at these to measure just some overall cell body diameters. We're going to revisit these slides later on in the semester and make some additional observations using all the knowledge that we learned over the course of the next few weeks. And that's going to help you practice your image skills, but also your microscopy observation skills as well. So that's all for this one, and I will show you some more cool tricks next week.